from the University of University.
No, the lab is common to both universities. So now when I
only one position.
Yes, I've got only one position, but that's enough for the moment. And I'm not on the CNRS neither, but yes.
And so she about models.
Okay, thank you. The guy told me to wait a little. Okay, it's okay. Okay, thank you very much. Okay, so yes, today I will talk to you about some models for biofilm growth. So I will try to explain to you what is biofilm growth, to give you just, I'm very bad with this kind of thing, okay? To give you an idea of some models that are existing for biofilms, and give you two applications, one for microalgae producing lipids, so basically biofuel, and a second application to gut microbiota, and I will tell you in a few seconds what is gut microbiota. So biofilm, maybe you don't know this word, and you are not aware that biofilms are existing, but for sure you have already encountered some of them. So here you have some, so there are not always nice picture of biofilm. Some, like, okay, dental plaque is a kind of biofilm. You can find some in industry, so this is not, you can find some biofilms in fontaines, so these green patterns that you can find in fontaine in reality are biofilms. And Pseudomonas aeruginosa looks nice, but in fact it's a terrible disease. So you have to be careful of biofilm, especially when they look nice. And what is biofilm? I have not told you what is biofilm exactly. So, no, it's not written here neither, but in fact it's a form for some biological organisms to live together, to cooperate, and to have a common way to live and to be stronger. So for a long time, biologists were considering that organisms were living on their own also because it was more easy to study and they have some more basic behavior. But now, from the 80s, it is far common to consider that, in fact, all biological organisms prefer to live together as we do. But it's also the case for bacteria and all and other things like that. So, how a biofilm is formed and is growing. So I try to find a simplified description of biofilms on internet. And this is a simplified version of biofilm for biologists. So a simplified version will take 8 steps, I think. So you have some bacteria, they are settling on the surface, they are losing their ability to move, they are creating this kind of shelter, which will be called the biofilm, which I will call biofilm in the end. So you need to have nutrients, of course, like every biological organism, they need nutrients to live. Okay, they give a kind of special microenvironment, but then there are some of these organisms who are having back their faculty to move, and they are going a bit further to disperse and to form a new colony. So that's basically the scheme of a biofilm formation and growth, and I will concentrate on these 8 simplified steps. I will concentrate on step 3. So I'll give you an oversimplified version of a biofilm growth. So I will assume that I have some bacteria on the surface, and I will try just to see how a biofilm is developing on the surface. So just only this part. I will not consider any diffusion of the biofilm further. No, okay. I think it will be a running gag during all the talk, but okay. So basically, what I will consider as a mathematician as some essential feature for a biofilm is that you have some microorganisms who live in interaction, who are anchored to a surface. They need to be attached to a surface, and they will produce an important thing, which is this extracellular matrix of polymeric substance, which I will call sometimes EPS, and which is this kind of glue part which provide a shelter for the organism to survive. And to have the formation of biofilm, it is often essential to have water and also to have nutrients, of course. So this is just to have a picture in mind. This nice, like, it looks a bit like this yellow flowers that you can find in spring in southern France, mimosa. I don't know the word in English. But in fact, it's a Staphylococcus Coccus aureus, that maybe you have heard about it, and it's not, it's not really nice. So you can see here the bacteria anchored to the surface, and you can see what EPS looks like. This is this kind of chewing gum thing that is around. And this is essential to the survival of the biofilm. So in this talk, I will consider 3 types of organism. This cyanobacteria on fountain walls. So this is a picture of fountain walls that I showed you before. And you have this cyanobacteria forming a biofilm and developing on a fountain. I will talk as an application to microalgae producing lipids. So here you have also a picture of microalgae, and I will just give you an idea later what it is. And I will talk also about gut microbiota, which are the bacteria that are in your colon, in your gut, in your digestive tract here. And we can also consider that they are living, forming a biofilm anchored to the wall of your colon. So you see that this is 3 very different types of organism. Of course, if I'm explaining to biologists that I'm considering that more or less they behave the same, they just don't agree. But as a mathematician, I will try to find a formalism that will enable me to write some models that have the same, that have some common feature and that have the same structure for all these 3 systems of organisms. So just to justify why I'm using partial differential equation, maybe the first point will be that it is my specialty, but I'm trying to be a bit more convincing. I will try to study, so I'm studying a system which depends at the same time of time and space, with a large numbers of organisms, they are in interaction, and quite often they also interact with their environment. So this is a scheme reproducing your colon, your digestive tract. And so you have these bacteria which are around, and there is you will see that there is an hydrodynamic flux in the center of your colon, and on the wall there are some mucus structures, that have to be taken into account, and your bacteria needs to survive into this wall environment, and it's quite important to have the flow and to have the mucus. So I will consider not only the bacteria, but also, at least for the 3rd application, an interaction with the fluid. And for all these reasons, so I'm considering a model at the macroscopic level and PDE. So what are the biofilm models that are existing? So biologists became aware of the importance of studying biofilm in the 80s. And of course, mathematical model took a little bit of time to appear, but since 2000, you can find some mathematical models in the literature. So the first type of model, for example, will be some agent-based model, which are quite simple to compute and to write. So you can fix the rules that you want. So here, for example, this is Pseudomonas aeruginosa biofilms, where you have some motile. So motile are the yellow one, and non-motile are the blue one bacteria, and you put them on the surface, and you're putting some rules when they should grow, when they should move, and how they would behave, and you can do a simulation like that. So this is a kind of simulation that is done by a team in Netherlands, in Delft. And of course, there are some what I found as problems with this kind of models, because if you want some realistic computation, it will cost quite a lot of time to be realistic, to have the right numbers of particles, and of course, this is high sensible to initial data. So now, another type of model that you can find is some model with some interface. So you're assuming that your biofilm is a green area here. You're assuming that the white area is a non-biofilm area. This is just liquid. And you're trying to compute the interface between these 2 regions, these 2 areas. And you will just put a law to compute the evolution of biomass components into the biofilm, into the green region. And you will also fix a rule to compute the evolution of the boundary through level set formula and through adapted algorithm to compute it. And you see that, for example, in this article, Albrecht and Clapper can compute the evolution of your biofilm. You can see that they can also take into account an active biomass part and an inert biomass part, the inert biomass part being on the back of the biofilm, and you see the active one, which is growing and which makes the biofilm grow. Okay, and if you don't want to use an interface, which is my case, I would say that this is a third kind of model that you can find. So I will try to. I will take a little time to explain it to you, because the model I will present you after, and the kind of model will be really based on this idea. So you consider that your biofilm is composed of phases. So let's say that you have living cells, that you have dead cells, that you have your extra polymeric substance, EPS, E, and liquid L. So this is 4. You have your 4 phases. And you're trying to write an evolution equation for these phases. And what you're considering is that, in the sense that you have described all the phases that are present in your model. So what you will compute are volume fraction. A volume fraction, you can see it as a percentage. So at a time t and at a point x, you are computing more or less the percentage of living cells that you have, of dead cells that you have, of EPS and liquid. So you will have an equation, which is not written on the slide, but which tells you that the sum of these 4 phases should be equal to 1, because you make the assumption that you have considered all what is in the biofilm and nothing is left. You have no void and no other phases present in your biofilm. So to describe the volume fraction, you will use some mass conservation equation. So you will write this mass conservation as a derivative of your volume fraction with respect to t, plus the divergence of the phase of the volume fraction b multiplied by the velocity. And you can write one equation for b, one equation for d, one equation for L. And you see that here I put some little dots, because for the moment I didn't want to write any source term, but the source term will tell you how each of your phases is growing, one with respect to the other, and how they interact one with another. And you can see here that there is some, so you have 4 equations, but you have the velocity v, and the velocity v has to be prescribed in some way. And to prescribe this velocity v, here in this article, Angwing, King, and Ward, and just close the system thanks to an additional condition, which was that the liquid phase is proportional to the quantity of EPS, which is not very well explained, and which has, so this is a first problem, but the other problem is that, okay, you have one condition, so you can compute your velocity in the 1D case, but if you are writing the same system in the 2D or 3D case, your velocity are becoming 2D, your velocity here is becoming a 2D or 3D vectors, so if you're in the 2D case, you will need another condition, and even the first one is not very convincing, so to add a second one is even harder, and the third one will be even worse. So we want to avoid to use such a condition to close our system, and to find a setting that will be able to write a system in 2D and 3D case. So this work began with a collaboration in Roma with Roberto Natalini, Cristiana Di Russo, and Fabrizio Clarelli. Okay, so there is, okay, I will try not to talk about my life, but the story was very nice at the beginning. It was to study this biofilm and fountains, and the first idea was to compare the fountains in Firenze, in Italy, with the fountains in Granada in Spain. So I was just expecting to do some very nice traveling in Italy and Spain, and of course this part failed, and what remains is a mathematical model. So, okay. So you see, I just end in the mountains taking picture of this cyanobacteria biofilms in the river in the summer, which is not that bad, but okay. So you see, so I want now to write the same kind of model, still without interface, to have some equations which are valid in 2D and 3D, and to take into account the few information I have about I had at the time about this cyanobacteria. And of course, they are doing some photosynthesis, so at a point, the influence of light should be also taken into account. So this is how it looks at the macroscopic level, and if you're looking to the cyanobacteria at the microscopic level, there are this kind of, okay, here they are. They have this shape. So I'm writing the same kind of equation as before. This is, okay. I'm writing exactly the same kind of equation as before, as I just told you. I have some volume fraction for cyanobacteria, for dead cyanobacteria, for EPS and liquid. And the slight difference that we are taking here with the previous model is that we are using two velocities. We will prescribe the velocity in a way, so we can have several velocities. We can have a solid velocities, Vs, which will be a common velocity for cell and EPS. And here it's quite, you can justify it because your bacteria are encapsulated into their EPS, so they will move at the same speed. And we will just add a velocity for the liquid phase, which are called VL. So I have these two velocities. And I still have the volume constant I explained to you, which makes that the sum of the volume fraction are equal to 1. Okay, so now I have to tell you several things about this system. I have to explain to you how to construct this source term, and I have to explain to you how to compute the velocity. So where is the biology of the system? The biology will be in the source term. I explained to you the source term is where I describe all the exchanges that take place in this system. So, for example, here, my bacteria are growing. So we have a birth term for bacteria where we have a product between liquid and bacteria because liquid is important for the bacteria to grow. And another important term here is EPS production. So the bacteria are producing EPS when they are in presence of liquid. So here again, I took the product between liquid and bacteria. And okay, for the moment, so I told you as a beautiful project failed, at the end we consider just the bacteria with very, very little information about it. So we took some very simple source term, and the idea was just in a sense to control that our model was giving something relevant before using it in more complex situations. And I won't describe exactly the influence of light, but here in the coefficient, you can make your coefficient depend on light with an optimal light. And this will be a bit complex because we will take into account the light absorption by the biofilm. So when I'm writing this E, in fact, this E is depending itself on the volume fraction BDEL. So this is something very nonlinear here, but I will skip this part. Okay, so I explained to you how to construct the source terms, so putting all the biology you know in it. And now to model the physics, the prescription of the velocity will follow some physical rules, and we will use some physical rules, and we will write a momentum conservation equation to be able to compute the velocity. So we're following some idea of mixture theories that have been developed by physicists like Raja Gopal and Tao. And this formalism of using a mixture theory were used a lot by Luigi Preziosi, who is here. So if you especially to construct some model for tumor growth. And we took this idea to adapt it to our system of biofilm growth. So basically, here again, in fact, to know the physical behavior of a biofilm is something very, very complex. The discussion I had with the physicists told me that they're trying to do some experiments, but on the same biofilm, you can have some elastic part and some viscous part for the same biofilm at the same time. So describing the physics of a biofilm is quite difficult. But okay, we need some equation to prescribe the velocity. So we're writing the velocity also with the simple forces that we can take. So we take an interaction forces between the phases. We take some constraint, but we take them as simple as possible, and an hydrostatic pressure, which is essential. So this pressure is essential because if you remember well, I have this constraint B plus D plus E plus L equal to 1. So I have a constraint that my system has to satisfy. And in fact, this pressure, like in Navier-Stokes, this pressure has the effect of a Lagrange multiplier so that the constraint is satisfied in the end. So we end up with this system. So this system is a little big. It's not so simple, but it will be even worse after. So for the moment, I just write, this is the only one I will write to because to the end of the talk, the system are becoming worse and worse. But okay, but it's not a problem for us because we have now a formalism. We are writing the equation of conservation of mass to describe the volume fraction. We are writing the equation of momentum conservation to prescribe the velocity. And we have an incompressibility constraint, which comes from the constraint of volume, the volume constraint. And that's the system. And we are able to, from that, so what are our contribution on this topic, on the cyanobacteria biofilms? The contribution that we made was first to write this system, to modeling the dependency on light, and I skip it, I have not shown it to you. And you see that the mathematical, so we did a numerical study, sorry. So we write a numerical scheme and we had some little numerical problems to solve. There was a part also was to estimate the parameters. So we don't have any data, so we estimate it from the literature, so what we can find on the cyanobacteria in the literature. And okay, we were able to give some simulation in 1D, 2D, 3D. And just to be precise, the analytical study of this system was done by Roberta Bianchini during a PhD with Roberto Natalini in 2016 for the 1D case and 2019 in the 2D case, meaning that it's not so obvious to make a mathematical analysis of this system. They use quite advanced tools for hyperbolic system in order to prove the existence of solution. Okay, so now I've got a setting and I've got a formalism and I can use it to a more complex situation. So the first application I want to mention is a work on microalgae biofilm. So microalgae are, there are some algae and they are considered as a nice alternative for a lot of things at the moment. So I don't know if in the end, in some years, we will eat algae, we will have algae in our motor and so on. But they are considered as a nice alternative because you have a lot of different species. They can grow, for example, in ocean, which are not very used. I mean, they are not in competition with agriculture, for example. And people expect them. So I think that yes, I'm sure you can already find some in alimentary. So it is not quite common, but in Brittany you can find some little algae things. You expect to use it in cosmetic, in water treatment, and in biofuel. And the project here was to use microalgae to produce biofuel, so to produce lipids. And okay, you can see this here, a small microalgae, and you can see this yellow dots. So these yellow dots are some lipids. So this is a microalga, okay, a cool microalga, but when you stress it, so stressing it means no nutrient. You are not feeding. You are not feeding it. So the microalga is starving, and when it's starving, it's producing lipids. So well, I'm not too fussy about making a microalga starve. So that seems quite okay with my conscience. And so you're trying in a way to stress the microalgae so that they produce lipids. And to be honest, for the moment, your car is not. You do not put any biofuel from microalgae in your car because the processus is not. So there are some prototypes, but for the moment, the processus cost, the energical cost of the processus is higher than the fuel that it gives in the end. So for the moment, it's not. The project. Well, I mean, maybe the positive way to say it is that there are still a lot of doing research. To have some nice microalgae producing biofuel. Okay, so to make them, so one way to gain on the energetical productivity is that usually the microalgae grow in a raceway, so this kind of big water tank where they can swim, and it is quite costly to harvest it and to make it dry. And one idea was to make biofilm growth on some ropes or on some tissues where you can take it easily. And the idea was to, of course, okay, so I'm coming back to the mathematical model. So, the idea was to derive a mathematical model that will give us the description of the growth of microalgal biofilm, and to try to estimate the production of lipids that it gives. And we will try to have something to optimize to have the highest possible production of lipids. So you see that here, so we keep the same kind of model. So I will have some phases, that I will call the compartments or components. So I will have some phases. They will not be the same as before. And here I was working with Olivier, maybe I put his name somewhere. We were working with Olivier Bernard, who is in INRAE, Sophia Antipolis, and also in the Observatoire de Villefranche near Nice. And he has a very good knowledge of the biology of biofilm. So in our source term, we can put a lot of things about how this microalgae grow and die and produce EPS. So basically, this is the summary of what is the biology we put in the model. So you can, so maybe I have to begin with the phases. So a biofilm will be composed of microalgae and will be composed of, so extracellular matrix is the same as the extrapolymeric substance that you have seen before, so this EPS part. And we will have a liquid part also. And we split the microalgae, in fact, in two components. We have the functional biomass, which are all the components that make the microalga live. So all is what is able to create lipid and so on. And we will take into account the carbon pool. This is where all the lipids will be stocked, where all the carbon will be stocked within the macroalgae. And since we are interested in studying lipids, it is quite important to have these two parts separated, because we are really interested in the carbon pool of the microalgae in the end with this model. So we have these four main components: carbon pool of microalgae, functional biomass of microalgae, extracellular matrix, and the liquid phase. And you can see that in the liquid phase, we consider also three that we call dissolved components. We consider that they do not have any volume, so they will not appear in the equation that I presented you before, but there will be also some equation for them. Also, they won't be on the volume constraint equation. So the dissolved components are carbon dioxide, oxygen, and nitrogen, which will act as a nutriment for the microalgae. And here we are looking at all the reactions that are present in our system. So mainly the microalgae are doing photosynthesis. So of course it is quite awful to try to read this diagram. I'm not expecting you to understand all of this, but okay. Once you are a bit used, you see here this is the arrow which will tells you how the photosynthesis is happening. So you have to take some liquid and some carbon dioxide, and it will produce oxygen and carbon pool. And you have also the respiration, which is the reverse reaction to photosynthesis, will happen also. So, for example, if you have too much oxygen, photosynthesis will not happen, but it will be respiration on the opposite, which will be present. And you have the production of functional biomass, which is important for the microalgae to be alive. And of course, we are interested a lot in also the production of extracellular matrix, because it will be full of lipids. So in the end, what we'll try to optimize is what is in the extracellular matrix and what is in the carbon pool. So you are writing all the reaction rates of this reaction. You can also take into account some limitation for this reaction. So for example, photosynthesis, of course, will be limited by light. So we will have some light-dependent coefficient to model this. The photosynthesis will be inhibited by oxygen. So if you have too much oxygen, you won't have any photosynthesis happen. And you can write your source term with all this information in it in a quite systematic way. So I do not showing you what it looks like in lag because it is quite big expression, but the way to find it, it's quite easy once you know, once you have all the information. So, we wrote these two terms from a chemical reaction with some saturation effect, inhibition effect. You have some threshold effect on the quota of functional biomass. You need to have enough functional biomass so that the microalgae is living. And you have sometimes also some optimal value effect. For example, the microalgae there is an optimal value of the light intensity. So if there's not enough light, they won't do any photosynthesis. But if there is too much light, they will be also inhibited because they are kind of blinded. So you can put all this in your model. We put some supplementary equation for nutrients. And we did quite the same numerical study, except that we had one or two more problems to solve. But as before, we can do some simulation in 1D and 2D. And here is what it looks like. So maybe it's not very simple to understand. I'm sure it's, but what I want to show you first is that you can compute, of course, a lot of things once you have designed your model like that. You can control the limiting factors, for example, for photosynthesis. They are here. The limiting factor for photosynthesis. You can compute all that to try to understand what's happening in your system. But the most striking curves are the ones which are here. So here we represent, you see the volume fraction of the four components that I mentioned. So the carbon pool, this is the total biofilm, algae and extra polymeric substrate, EPS. We have the fraction of EPS and the fraction of functional biomass, and the rest, which the liquid is not represented because it is the rest of the system. And so what is a 1D microalga biofilm? Well, this is a biofilm which is developing in height. So here, the abscissa is only the eighth of my domain, and here what I'm representing is some kind of percentage of all of these components. So here you have the evolution. We have the evolution of the system. I present here a case where the light intensity is constant, but you can make some light variation too. And you can see that a bit like the computation that I presented from the article from Alvis and Clapper, you have a front. So you have a front which is growing. The biofilm is growing as a front, and you can see the composition. You can see also point by point the composition of the biofilm. And what we notice is that the living part of the biofilm is on the top. That the EPS is more present in the front of in the bottom of the biofilm. So okay, so this is the kind of simulation that we can make, and we can estimate with this kind of okay, this is the kind of simulation that you obtain in 2D with the same conclusion. In fact, so this is the whole biofilm, and you can see. So this is a time t equal 5 days, and then t equal 20 days. So you have the evolution of a biofilm. And you can see that this is the ratio microalgae versus the whole biofilm, which tells you that in red, this is the living part of the biofilm, the part that makes the biofilm grow and live. And you can see that it is present on the front, more on the front than on the bottom of your domain. And you can see here also the representation of the oxygen. So the representation of the substrate, which tell you that more or less what is making the biofilm grow. I mean, it's not only the only feature, but when you do not have any more substrate because the microalgae consume all the substrate, the biofilm will grow to look for substrate on the top of the domain to feed the biofilm. Okay, and that's it. And so the end of the project, which was concluded and which was accepted in an article just last week, I think, was from this 2D simulation to try to estimate some daily production rates, some daily lipid production rates, and in a context of harvesting. So we are making our biofilm grow, and we are harvesting it. So we can also try different shape of blade. How do Do you want to harvest it with some kind of kernels, or do you want to harvest it just like cutting it, and so on? And you can try to optimize recording according to the harvesting rate and harvesting period. So it's not so simple because, as I just shown you, the living part of the biofilm is on the top. So if you're just harvesting too much, your biofilm, in a sense, will die. You won't have any more living microalgae, and you will be full of lipids, but your biofilm will be dying. So there is a sort of compromise to harvest, not too often, because your biofilm needs to, after harvesting, to have time to be alive again, and not too and maybe not too deep, not to kill other biofilm. So the conclusion is that okay. So this is there is no optimization techniques under this graph. They are very simple graphs. We just make some simulation and plot it pixel by pixel. So there is nothing clever about it. Okay, but it was just to see if it was an interesting question to try to do some optimization on what we can obtain. And we can see that we obtain very different results with respect to what you want to optimize. If you want to optimize the full biofilm, you obtain this kind of picture. And if you want to optimize only the extra, the EPS part, you will obtain this kind of picture. And if you want to have only the microalgae, so let's concentrate on the microalgae. If you want to have a lot of microalgae, you see that you need to wait a bit of time, around 1 week to harvest, and but you can go quite deep to harvest it without not too many problems if you are patient enough. Okay, so we have a model which can produce some 2D simulation and which you can use to try to optimize the production of your microalgae biofilm and your lipid production. Okay, so the conclusion is that from the first model, what we had several ingredients here. The structure of the model is the same. The number of equations is a bit larger because we have all the dissolved components and the nutrients that are taken into account. And we were very, very more careful and on the source terms, they were very more developed because we have many more biological information. And I want now to talk about a second application, which is also a model to describe the gut microbiota. So the gut microbiota is so this is a picture of your colon, everybody's colon, and you can so maybe it's not very you cannot read very well, but just to sum up, you have a lot of bacteria in your colon, and they are responsible for a lot of things. In fact, they are doing a lot of things. Those bacteria in your colon are doing a lot of things. They are responsible of many disorders. Now we know that even so metabolic disorders like obesity, of course, but inflammatory or even mental disease can be affected by your microbiota. You get microbiota. Their principal role is the body hydration. So from what we eat, the bacteria are extracting water. They are pumping water through the walls and send it to the whole body. And they have a role also of control of pathogens. So as I told you before, in your colon, which is here, you have a mucus part, and this mucus part, in fact, has two distinct parts. There is one part which is favorable to bacteria, and your microbiota live in this favorable environment, and so this mucus plays the role of the EPS in a sense for this microbiota. That's why this gut microbiota system can be seen as a biofilm, as a kind of biofilm. And you have a second layer which protects your all your body from pathogens and from which inhibits this bacteria to pass in your body. So this microbial community is therefore responsible for digestion, control of pathogen, and regulate your immune system. So now you have an important study in microbiology of the behavior of gut microbiota. So the aim of the model that I'm presenting you is to describe microbiota in its physical environment. And so the physical environment is the mucus on the wall. And also you have, of course, in your, as you can imagine, in your colon, an hydrodynamic flux, which is here. And your bacteria, to remain, your gut microbiota, your gut bacteria, to remain in the colon, they need to overcome this flow which is passing in your colon. So the model will include a metabolic description of the digestion. So this was have been already done in 2020 by a model from Munoz Tamayo and Beatrice Laroche and so on. So you have the expression of the digestion processes in the colon. We will add some hydrodynamical description of what's happening in your colon. So there were some previous models that were describing your colon as compartments, as several compartments. And here we will use a continuous model, and we will just model the colon as a cylinder, as a long cylinder. And we want also to describe some feature of the bacteria, spatial behavior. So the bacteria are performing chemotaxis. That is to say that they will be attracted by some of the components that are present in your colon. You also have, this should not be here, but okay, anyway. Peristalsis is not a bacteria spatial behavior. Peristalsis is just the contraction, so I'm sure that you have already feel it also. When you're in the digestive time, so this is not the case now, but in a few hours maybe you will think of this talk, you have some contraction of your intestine, of your colon. They always happen, and you have this peristaltis, which has the role to mix what is in your colon, and especially the bacteria and the flow. So this is the metabolic description. Once again, I won't comment. I will not comment this system too much. You can just to tell you that there are 13 reactions, and of course. You cannot read that because you don't know what are all these signs. But okay, this is liquid. This is the mucus that you have on the wall. You have some polysaccharides, which are more or less what's arriving in your colon from what you have from your lunch. So this is what's remaining from your lunch, which is arriving in your colon. And you have some bacteria, so this is the gut microbiota itself, which have the role of catalysis for all this metabolic reaction. So there is a transformation of polysaccharides in monosaccharides, from monosaccharides to lactate, acetate, propionate, butate, and so on. And all these short chain SCFA, short chain fatty acids, are just then sent into your body for the just for you to be well.
So this is the...
But okay, so more or less here the components of our model, there will be 8 components. So as a mathematician, it's not a problem. I'm just keeping the equation that I have, and I'm writing 8 equations instead of 4. And I'm writing also some equations for the diffusing solutes as before. So okay. See, no, I wrote some equation. I did not dare to write you the whole system because anywhere on the slide there won't be enough space. But I just write this toy model systems. So okay, I have to make some advertising here. So this is a work from SEMRAX 2015, I imagine, because it was or 14, I cannot remember. But this was a work in SEMRAX. I don't know if you heard about SEMRAX, I imagine yes. So this is a nice summer school which takes place here every summer. So you can spend 6 weeks, for PhD students, you can spend 6 weeks here, and it's really a nice, a fantastic experience, I should say. So there will be a nice SEMRAX this summer about transport equation, and I just recommend you to do all the SEMRAX that you can do, because it's super nice. Okay, this is the end of that advertising. I'm coming back to my equation. Sorry about that, but at a point I have to talk about equations. So here, so in the SEMRAX, okay, I will take the story back. So we began to write this metabolic equation on my colon, which was a cylinder with some a prescribed fluid velocity. And it didn't work at all. I mean, the bacteria were just all sent out of the gut of the colon, and we cannot see anything. So the idea of the SEMRAX was just to construct a small, very small model with mucus volume fraction liquid volume fraction, and try to understand how to prescribe the velocity. So here we wanted something more simple that I just tell you before, but you can see that the structure of this equation are exactly the same as before. And the idea was that the flux was given by the flow that is in the middle of your column. And we wanted to write a Stokes equation, but the viscosity of the Stokes equation was quite important because your bacteria were remaining on the wall, near the wall where the mucus were, and were not in the central part of your column where the fluid was passing. And in fact, what was tested into this MRAX and what was the key ingredient for the full model after was to have, for the Stokes equation, so which gives the velocity, a viscosity which depends on the volume phase of the mucus. So you have a viscosity which depends on space, but not directly on space. It depends on space through a dependency with a mucus dependency. So once this toy model has been tested and approved, you can write, I tried to write it, but okay. You can try to write your equation. So I think I'm running out of time. So I'm just saying that rewriting the same kind of equation as before, we are adding all the ingredients that we have now, that is to say on the boundaries, we are adding this contraction, which are peristalsis, water pumping, and so on. So everything you want to describe. And we were able to perform some simulation of the evolution of the gut microbiota, making also some asymptotic model because we consider that colon is much more longer than it is wide. And we were doing mostly the same kind of situation, and we were able to produce. So I'm getting far, but we were able to study the spatial structure, the spatial distribution of the gut microbiota. So here, for example, the mucus, we were able to show that the bacteria were remaining with this setting, were remaining in the gut. And we were able to show what is the distribution. And one thing that we're expecting to do, and which will be one of the feature of a PhD thesis that will begin in October with Pagel, who is here. So that's why. Yes, don't be too shy, Pagel. Will be, for example, to study. Well, one point, for example, will be to study the competition between some bacteria in the gut. So you have bacteria with several features. If we put a bacteria which is very strong in chemotaxis, so which knows how to go to take food, and one which is very good in metabolism, that is to say, once the food is here, it's very efficient to grow, which one will win? And so some first simulation tells that it depends on the coefficient. So this will be quite nice to study this kind of thing. Now that the model is defined. Thank you very much.
I'm not really into modeling, but from a very big question, why you do not have any?
So in the original model, the idea was just to have the propagation of front through hyperbolic equation, and to see a front of hyperbolic equation. And we wanted a very clear front between a void region and a region with biofilm. But then in the end, talking with biologists, I mean, the first was a very toy model that we do within mathematicians. And when we talk with biologists, in the end, they just tell us, no, you cannot, you have to put some diffusion. And you can see that in our last model, in the end, we end up with some very small diffusion coefficient because, yes. Yes, or we didn't try to do the analysis with 8 components and so on, but yes, it's probably simpler. Yes.
And another question is just wondering, is it possible to problem average? Because you have two, but also
yes. No. You won't be able to, except in the 1D case. In the 1D case, you can integrate your incompressibility equation, and so you can. Okay, sorry, I'm very bad with this. Definitely, I will need to have some lessons about this. Okay, in the 1D case, you can integrate this equation, and using boundary condition, you can express one velocity with respect to the other. But in the 2D and 3D case, you are unable to do it. So you cannot express one velocity with respect to the other.
Very last. Very short.
I expect a
I was also wondering, how can you describe mathematics, some of your fractions?
Through the pressure. The pressure is a kind of Lagrange multiplier. So in some sense, the existence of this pressure, you can write an equation of the pressure, which recalls you in the sense that you have this constraint. Yeah, exactly. Yeah, that's it. Except that the velocity is the average velocity, but it's exactly the equation. Yes, that's it.
Thank you very much.
You're welcome. I think it's time to go to eat, to make some little experiments about gut microbiota. That's it.